Okay, hopefully today's YouTube lecture will reinforce what you started to learn yesterday and doing some of these bigger, more complex Punnett squares. We go back to Punnett squares after meiosis because we're using the concepts we learned in meiosis, the idea of the splitting of the chromosomes. So an individual who has 46 chromosomes in their body cells, those 46 chromosomes will split and only 23 of them will end up in a sperm or in an egg, which will then fertilize a sperm or an egg with another 23 to go back to 46 chromosomes in the baby. So this idea of the splitting of the chromosomes, we want to carry through to the idea of the two-factor or the more complex Punnett squares. If we go back to the one-factor Punnett square, I'll just put a 1F here, one factor. So for example, if we had a pea plant that was homozygous tall and we crossed that with a pea plant that is homozygous short, we do the Punnett square this way, right? So if you just look at what I'm doing, what I did at that point there, in essence, what I'm doing, I'm saying here's mom, who's got two T's, big T, and what I'm saying is by putting one T here, I'm indicating that when this mom makes an egg, she's only putting one of those T's in that egg. So what is this egg going to do? This egg is going to combine with a sperm. So on this side of the Punnett square, if we put dad's sperm, okay, I don't want that yellow. If I put his little T's on the left, and that's representing his sperm. So if I think about dad's got two T's, right? And this, I'm putting one T here, indicates that I'm taking those two T's and I'm only putting one of them here in his sperm. This way, when I put the offspring back together again, in here, I put mom's egg together with dad's sperm and I go back to the original chromosome number. Here, I'm just modeling two chromosomes. So again, look at what we did here. In essence, we just did meiosis and fertilization. I'll change colors one more time here. So I have two chromosomes here, and when I make the egg, I only put one in it. I have two chromosomes here. When I make the sperm, I only put one in it. 1 plus 1 goes back to 2 in the offspring. Now we want to take this idea and expand it to two factors. Let's take that same thinking, but now I'm not just a tall pea plant, but I'm also a green pea plant. So when we look at two factors, instead of indicating 2 letters, we indicate 4 letters. So this would be one genotype of a tall and green pea plant, if I assume green is dominant over, we'll say, yellow peas. Another possibility would be this, homozygous tall, homozygous green. Another possibility would be this. This would be a tall pea plant with yellow pods, yellow peas, if we assume yellow is recessive to green. We could do this. There's a number of, here's a homozygous short, homozygous yellow pea plant. There are, I believe, 9 different genotypes when you looked at two factors at the same time. So how do we do a Punnett square like this? Look at this individual again. The heterozygous tall, heterozygous green pea plant. Again, hetero meaning different. So the T's are different and the G's are different. So we heterozygous tall and heterozygous green. So if we imagine these letters represent genes that exist on chromosomes. In essence, what you have here is 4 chromosomes, or 2 pair. Let's go back to meiosis. When this individual makes a sperm cell, are they going to put all 4 chromosomes in the sperm? No. How many are they going to put? Half, right? So 4 in the parent becomes 2 in the sex cell, or in the gamete. So, well, which 2 am I going to put? So the way meiosis occurs is each pair of chromosomes gets shuffled so that one member of each pair is what ends up in the sperm cell. What that means is one of the T's and one of the G's. You're not going to end up with a sperm that has a big T and a little t and no G's. each pair will donate one member of that pair to the sex cell. And that's how just the shuffling during meiosis occurs. It's possible to generate a sperm that has a chromosome with tall and green in it. But it is possible, instead of the big T and the big G, that a sperm ends up with a little t and a big G. So hopefully you see the next options here. It's also possible that a big T ends up in the sperm, the big T chromosome ends up with the big G chromosome, or it's possible that the little t and the little g end up in the same sperm. So what we're modeling here with the letters is the traits that are determined by these genes. So we have tall and green, short and green. I made a mistake here, right? This should be tall and yellow, or short and yellow. These are the 4 possible different kinds of sperm that this individual can generate. Or if it was a woman, these would be eggs. Now, these in essence are the possible rows or columns, depending if you put this individual on the top or the left of the Punnett square, for my Punnett square. Let me go back to the one factor just for a second here. So when I do something like this, right, and I generate a Punnett square that shows a big T or a little t, that's why I have 2 columns. I have 2 columns to generate the 2 possibilities of this individual, either the big T or the little t. If I go back to what we call the dihybrid, double heterozygote, the dihybrid. So when this individual makes a Punnett square, if I put his gametes on the top, he's going to generate 4 columns. Remember the options here, the choices here. He might generate a sex cell with a big T and a big G, or a big T and a little g, or a little t and a big G, or a little t and a little g. Well, what do I put over here in the rows? That depends on what other pea plant this one crosses itself with. In the Punnett square with the most possibilities, that would be an individual who on this side of the Punnett square is also, we would say, big T, big G, little g. And then this individual will also generate 4 different possible sex cells, or what will get you 4 different rows. This is the Punnett square that was gone over in the Ed puzzle yesterday. It was called a dihybrid cross. Dihybrid. So we should remember that term. This is a dihybrid cross. Dihybrid cross is a 4x4 Punnett square. You generate 16 different genotypes here, different, I guess, combinations, different boxes, but there will be 4 different phenotypes. So let me just put some of these together. So when you put the alleles back together, you put the sperm and the egg back together, you're going to go back to the 4 chromosomes you had in the parent. So if I just look in this box here, I would put my T's together, big T, and my G's together, big G. If I go over to this box over here, this would be a big T with a little t and a little g with a little g. This one here in the lower right would be a little t and a little t and a little g and a little g. Again, you're putting together the column and the row to get the result in the Punnett square. The column and the row. Column and the row. The results from this, if I can stay over here on the left, and let me erase this and get this out of the way. So the results in this Punnett square, when you look at them, you'll have certain offspring that are tall and green, certain offspring that are tall and yellow, some offspring that are short and green, and some offspring that are short and yellow. Now the assignment that I'm going to include with this is a Punnett square modeled like this. The phenotypes or the traits are a little bit different, but it is going to represent a pea plant cross. And you're going to, what I would like you to do is to color. There's a little key at the bottom with buttons. I'd like you to color those in 4 separate colors, and then color in the 16 boxes according to the appropriate key. So for example, if I want to use green for this, tall and green. So if I want to represent a box that's tall and green, well, this box is tall and green. Now I'm not going to fill all these in, but this one, this one would also be tall and green. This one would be tall and green. This one would be tall and green. So you're gonna continue that with the rest of the Punnett square. So tall and yellow. So let's do red. Tall and yellow. Let's see, here we go. Tall, this plant is tall and yellow. Yellow being recessive in this example. I think in your homework example, yellow is dominant. So don't get confused by that. Short and green, I don't have a short and green one. And then let's go do short and yellow. This one is short and yellow. And then in front of those, after you color those in, you're just going to count up the number of boxes. I'll give you a hint. What you're going to find when you do a dihybrid Punnett square, and again, if you pay attention to Edpuzzle, the results here will be the same. The number of individuals, offspring, that are dominant and dominant, in this case tall being dominant to short and green being dominant to yellow, is going to be 9 out of the 16 boxes. Extend just a smidge more before I let you go here. With a two-factor Punnett square, it doesn't always have to be a 4x4 Punnett square. So we mentioned this individual, right? This individual can make a sex cell with a big T and a big G, or a big T and a little g, or a little t and a big G, or a little t and a little g, right? So we generate, say, 4 columns again. But what if this pea plant is crossed with a pea plant that is little t, little t, little g, little g? Well, I'm going to show you a little trick here. If you look at this individual, so what do we say? We said this is what we're saying is the egg then. In the egg for this individual, we're going to split these 4 chromosomes into 2. In the sex cell, put 1 of the T's and 1 of the G's. Well, if you look at the T's, they're both little t. They're both the same T. Whether you pick the chromosome on the left, the T on the left, or the T on the right, they're little t. So really, there's only one possibility for T. It has to be a little t. And now look at your G's. They're both small g's. Whether I pick the G on the left or the G on the right, either way, it's going to be a small g. So this Punnett square, if you look at it, it's actually a 4x1 Punnett square. So on the top, yes, there are 4 columns, because I guess we're saying the dad here, because these are sperm, generates 4 different kinds of sperm, but the mom only generates 1 kind of egg. So when you do the two-factor crosses, the size of your Punnett square can vary depending on the possible gametes. So 4x4 would be your biggest possible Punnett square. 1x1 actually would be your smallest. All right, so we'll talk about this more in our next lesson.